Okay, hello everyone. Welcome to the Department of Psychiatry and Behavioral Sciences Grand Round Series, 2020-2021. I wanted to say just a couple of comments about our series. This is a new effort for this academic year, and we've converted Grand Rounds to a remote series, and this year have 4 advisory panels and also have input from psychiatry residents on covering major areas within psychiatry on education, clinical updates, staff, trainee, faculty development, and research. And we're aiming to have presentations covering all of these areas. We have several funding sources to acknowledge for the Grand Round series. And I wanted to again acknowledge Samhar, who's been coordinating a lot of the important details for Grand Rounds. One point is that we were able to get started with this series pretty soon after the idea came up to convert to this remote series. And I think that had some advantages where we could bring presentations more quickly to our department. It's also led to us finding some areas to continue to make improvements in. And one of those is in our post-presentation evaluation. We have a new link and a new evaluation survey to complete today. And that survey, I think, will make it more feasible for us to quickly review and synthesize the comments and get a summary of those comments to the presenters. So there's a new link today, and look for that after the presentation. And I'll move on to today's presenter. Today, Dr. David Oslin is presenting. And Dr. Oslin is a professor of psychiatry at the Philadelphia VA Medical Center and at the University of Pennsylvania Medical Center. Dr. Oslin also directs the MyRec in the VISN-4, which focuses on supporting research on implementing precision mental health care, which is actually what we invited Dr. Oslin to present on today. Today we'll hear about current evidence for precision mental health care and the methods of a new study called the PrimeCare research study, which is a blend of clinical trials and implementation science related to precision healthcare. So I'll stop sharing, and Dr. Oslin.
Great. Oops, where'd it go? There it is. And I'll start sharing. So let me know if that's showing, Joe.
That's showing.
Okay, great. So thank you, and thanks, friends. It was great to see Jürgen and Sir Murray in the audience as well. I mean, to whatever that means. I saw your name on the list, I guess, Murray. It's nice to be with you guys today, and even though we're thousands of miles apart. So I'm going to do 2 things for you today, and hopefully to generate some questions. I'm going to talk a little bit about the state of affairs with pharmacogenetics, and then just tease you a little bit with a trial that the VA is doing in this area. And let's see if there we go. Some disclosures. Janssen Pharmaceuticals is actually the PrimeCare study has contributed in-kind support, some in-kind support to that project, as well as Myriad Genetics. And I take grant money from a lot of different people. So I can talk about the relationship with those 2 pharmaceutical companies if people want to ask about it. My personal feeling is that they have no access to the data, and they had no participation in the design of the trial. It's been kept very, very separate, both companies. So this is a definition of pharmacogenetics from NIH. The idea is that genetic information or information about the genome can be used to help select medications or doses of medications that work for a particular person. I think it's a decent definition. You know, some people will say we need a definition for pharmacogenomics now, not just genomics, that there's other pieces to it, and the genetics is just one, but it gives us a sense of what we're talking about as a field. I want you to read this and ponder over it a minute. So this question will probably change in the next 5 years, but so for antidepressant prescribing, which of the following best represents how pharmacogenetic testing might be helpful? And I would just add in 2020 or 21. So it's helpful in selecting the most efficacious medicine. It's helpful in selecting a medication that will have less side effects. It's helpful in providing information on metabolism of selected medicine, or there's no evidence that it's helpful at all. So I'm not going to answer that right now. Hopefully I'll answer this by the end of the talk. And if you don't think I did, ask me the question at the end. As sort of a primer, there's really two aspects to pharmacogenetics. One would be thinking about disease genetics. And this is what most of us, when I went to medical school, learned about. So a particular disease may be kind of subtyped or divided up by different ways of thinking about understanding genetics. And some of them, like Huntington's, you sort of can't have the disease if you don't have the gene. But also there's cancer profiling, there's things like Alzheimer's. There's a host of disease-related genetics. And there is a component of pharmacogenetics that might say, if you know the right disease, you could pick the right treatment. That's not totally pharmacogenetics. We would call that disease genetics, not pharmacogenetics. Within the pharmacogenetics domain, there are really two ways of thinking about the utility of genetics. One is around the kinetics. How is that medication metabolized? So this is, how does your genes interact with the medication or the treatment, particularly around metabolism. The other would be pharmacodynamics. How does the genome say something about whether or not that medication works? I will tell you that we have almost no pharmacodynamic examples in our field right now, and like 99% of this is about pharmacokinetics. So that's a little bit, we're going to talk a little bit about that in some more detail as we go. Conceptually, this is the idea. When I go to pick a medication for a given disease for a given patient, there's a lot of things I think about in picking that medication. And this is assuming that we've made a diagnosis. So I have a patient that has major depressive disorder, and first time that I'm going to choose a medication over psychotherapy. These bubbles here are things that go through my head, go through my thinking when I'm going to select which medicine. And the question is, does pharmacogenetics or pharmacogenomics, is it another one of the pieces to this decision tree? This is an important concept because pharmacogenetics is not the only piece of the decision tree, and it probably won't be, right? So there's going to be times that you might choose a medicine that the genetics may steer you in a different direction, but there's other competing factors. So it's not a total black and white that pharmacogenetics will be the only factor that may influence your decision of medications. In our field, we have lots and lots of choices of medications for given disorders. There's lots of antidepressants, there's lots of antipsychotics, there's lots of benzodiazepines. So in our field, it is about choice of medicine as much as anything, as opposed to a field where there's only one treatment, and it might be about whether you use that one treatment or choose some other avenue of treatment. So why might pharmacogenetics be actually helpful to us? Well, this should be old hat for you guys, but first line antidepressant response, 50% is very much in the high end of first line response. It's typically around 30%. There's evidence that lots of antidepressants are never refilled. Lots of evidence that side effects are prominent for many people, and the reason that they don't stay on medicines or fail medicines. And every successful, I think for us in psychiatry, every time we try a new medicine, we lose patients. They drop out of care, they get frustrated with us or whatever. So if pharmacogenetics as a principle were effective to help us select medicines quicker to get to either the right medicine or medicine that's not going to be tolerated for somebody, all of that has huge public health benefit. So if I can take one treatment trial away from 70% of patients, that has profound public health cost and benefit outcomes. So that's sort of the concept here. So why is this not a science discussion, and why are we even talking about implementation and dissemination? It's because you can get these tests now from dozens of companies. The sky has fallen, and this is a multi-billion-dollar industry. And so I'm sure that you have all had patients come to you with their own test results, or come to you and ask you to get them tested, or you've had company representatives come to you or knock on your office and say, why aren't you doing this, and so forth. So there are literally probably 24 or close to 24 different companies that offer some form of pharmacogenetic testing. Interestingly, for us in psychiatry, most of them focused on psychiatry as a principal arm of who they go after, and that's because we prescribe, not we as a broad field prescribe many psychotropics. So psychotropics are number one or number two of the most prescribed medicines in this country. And so most of these panels really have a psychotropic focus to them. Now, interestingly, these companies have all gone after multiple genes that are on their panels. They've all decided to do panels of genes rather than giving you a single gene for a single drug kind of thing. And they bundle these together. Most of them use about 8 genotypes. I'm going to show you an example of that in a minute. And I'll show you what those genes are, but they're genes that if you're in the genotyping genetics area for a living, you will recognize most of them. A very important part of this is none of these companies are FDA regulated. So it makes sense when you think about it, but when you order a hemoglobin test or a cholesterol test, that is not regulated by the FDA. That is regulated by the lab. The lab is providing you a test result, and what we are doing is the process of lab approval is to make sure that you're comfortable that the test result that you got met some standards in terms of getting it. The labs that do pharmacogenetic testing do the exact same thing. They have the exact same rules that your local lab or LabCorp or whoever you use uses. They are beholden to give you exactly what you ordered and to give you high quality output in terms of the genotyping. The major difference between pharmacogenetic testing and ordering a cholesterol, let's say, is that when you get a cholesterol, it comes back with a reference range. And you as a provider, I would guarantee you probably 99.9% of you would understand how to interpret that cholesterol level. If it's 220 and the range is to 180, you'll know what a 220 means. I would also venture to guess that probably less than 5% of you, if you got back these 8 genotypes, would be able to interpret how to use those genotypes. So while you might get the exact genotype back, you wouldn't know how to go about understanding what it means for picking a medication for that particular patient. And so unlike the cholesterol example, for genotype testing, what you're really relying on them, on these companies for, is their interpretation of the genotype, not actually the genotype. So you can feel really comfortable that you will send, if you send a sample to 5 of these different companies, you'll probably get, most likely you'll get the same genotype back. They do use a slightly different methodology in terms of different SNPs and stuff sometimes. So you might not, there might not be 100%, but it's going to be close to that. What you will get back, though, that could be different is the interpretation of those genotypes, because there's not a standard right now in the field for how to interpret the genotype related to a particular medicine and a particular recommendation around that medicine. And the FDA does not currently regulate the space. There was some scuttle this summer. The FDA, sent out warning letters to all these companies and said, hey, you're doing something that we don't appreciate. They all pushed back. They won. The FDA didn't, and we are still left with companies not having a standard algorithm to follow to interpret genotypes. So it's just something important, an important piece of knowing this, because we rely on the interpretation, not the actual genetic test result. So this is an example of the genes that are included. So this on the left of the pharmacokinetic ones, these are all actually pretty standard. You'll see that they're all metabolism related, and most of them are the cytochrome P450 system. So what we're really talking about here when we're talking about pharmacogenetics in 2020 is are medications metabolized differently for this individual patient? And that is the full crux of that. And then the question is, if we know that somebody metabolizes drug A differently, does that help us either in choosing the medication or in dosing that medication differently? And I'll show you some clinical trial results about how this might be useful. You will see this is actually one of the company's panel of genes that they include in their testing in their portfolio. You will see that they toss in, this particular company tosses in 5 genes that they call pharmacodynamic. You'll also, if you know anything about the field, realize that these are pretty marginal, and maybe they are helpful, but probably not. And so really the meat of the story that we're getting from these companies is really around the pharmacokinetic markers that they're testing. And then just to flesh this out a little bit more, this is sort of what we're looking at in a particular patient. Is the FDA does actually label many medications as being in one of these 4 categories. So they might say that drug Y for 2D6, if you have this allele for 2D6, that patient will be a poor metabolizer. Theoretically, what that means is that person, if they're a poor metabolizer on the far right, that they will end up with a standard dose, they will end up with more medication on board than the normal metabolizer, if you will. And we know that for us in psychiatry, that will it probably doesn't change the efficacy of the drug, but certainly exposes the person to many more side effects. So most of our medicine side effects are very dose dependent. On the flip end is the ultra rapid metabolizer. And again, theoretically, that patient with a standard dose, if they're an ultra rapid metabolizer for that particular medicine, would end up with a serum level or therapeutic level that's markedly lower than what we're trying to achieve. Now, here's the other sort of fallacy in this field and where we are. As most of you know, our dosing windows are not very stringent. And what we really don't know here, and these companies are not beholden to this, because remember, they're only providing you a lab test result. They're not providing you actually clinical advice. What we don't know is, I'll use the example of sertraline. So let's say sertraline, a normal dose would be 100 milligrams. So in the ultra rapid metabolizer, am I talking that I really need to dose to 125 milligrams or 600 milligrams? How much ultra rapid are we talking about? And on the flip side, for the poor metabolizer, if I wanted to get an equal sort of a serum dose as a normal metabolizer, am I talking about 5 milligrams or am I talking about 95 milligrams? And this we don't know. So we feel very, very confident in these categorizations, but we don't really know the clinical interpretation of how it affects the dosing for that individual. And that'll be important as you understand how these trials have been done, which we're going to go to next. So this is the entirety of the state of the evidence. There are a whole bunch of 20 patient, non-randomized small trials, almost all done by companies. And the meat of the evidence is really at this point, I don't know, a couple days ago or a week or 2 ago, are 4 randomized, reasonably sized trials. And what you'll see here is they range in size from 280 subjects up to 1,100 subjects. Each one of them has gone with this concept of a panel of genes that they've looked at by using a commercial product. So they've not sent it off to your local lab to get 2D6 markers back. They've sent it off to one of these, in this case, each one of these studies is a different company. And they've relied on that company's interpretation of the genotyping. It's actually pretty straightforward to send to your local lab and get the genotyping done. The real informatics piece here is doing the interpretation. So we have 4 trials. I'm going to walk you through one of them, and I'm not going to tell you about the other 3 at this moment. But so the one I'm going to talk to you about is the largest of these, the trial that's called the guided trial, which was done by John Greedon at UMich. And it's a multi-site trial, 60 sites. They use what was then the company was named Assurex. Now they're Myriad Genetics that got bought out. And the battery that is sold under that company is called GeneSight. And this is probably the highest penetration and the highest market in terms of the market. So it's probably the one that has the most name recognition, the GeneSight battery. So again, they did about 1,200 patients. To get into the trial, the patients had to have MDD. They had to have failed at least one 8-week trial of an antidepressant in the current episode. So they essentially were being treated with an antidepressant coming into the trial, but still symptomatic. They were randomly assigned in the trial to either the provider got back the pharmacogenetic results, or they didn't get back the pharmacogenetic results, with the idea that the pharmacogenetics results would help the provider decide about dosing changes. Interestingly, in this trial, they did not give those results to the patient. And that's interesting because there probably is a fairly large placebo effect from getting a piece of paper that says this might be the right medicine from you, which is how the patients perceive it. The outcomes were evaluated over 12 weeks, and they had primary outcomes that related to was our main effect, and secondary outcomes that had to do with congruence or incongruence of the results. And I'll come to that in a second. So this is, if you've never seen an Assurex panel, this is sort of how the results are presented back to the patient or the provider. Assurex divides all the medicines. So remember that Assurex has actually 12 genes on their panel that they're interpreting the results for you for. So this is about a 20-page report if you've never gotten one. The genotypes are provided to you. So you will get the actual raw data like a lab report, like that cholesterol level. But at the beginning of it, they actually give you the interpretation. And they've made it, one of the reasons they're the industry market leader in terms of sales, not necessarily in terms of efficacy, is that they've made the interpretation of their report incredibly simple. So they've simply taken the medicines, cross-referenced those with the genotypes, and told you as a provider that you can use this medicine as directed, meaning that there's no known pharmacokinetic difference in that particular patient for these particular medicines. So in this example, most antidepressants are not effective for this sample patient. On the flip side is the folks in the red bin, and in this example, paroxetine for this patient. In this example, paroxetine is highly genetically influenced in its metabolism. And I honestly forget what the little footnotes here, the 4 and the 6 tell you what direction that is. I don't remember which direction off the top of my head, but it's either, in this case, it would be either an ultra rapid metabolizer of paroxetine or a poor metabolizer of paroxetine. So and then there's some that are sort of falling in between, meaning that there probably are some genetic influences to metabolism, but they're not as solid. So there's a bunch of different ways this report can be interpreted. One would be that these medicines, in this case, paroxetine, doesn't work for that patient. Well, that's actually, there's nothing in this report that says that. All this report says is that paroxetine is heavily influenced in its metabolism. And if you're going to pick paroxetine, you should be careful about how you dose. You either need to dose at higher doses or lower doses, depending on the direction. I will tell you that patients see this, and if you prescribe paroxetine, they're asking you why are you prescribing a red drug to me? It doesn't red mean bad. So the patients will see this report and say, why don't you give me one of these things in the green box? Even though it doesn't tell you that the green box ones will actually work better for that patient. They just tell you that we don't know of any metabolic differences basically in those medicines. The trials have all been set up. So those 4 trials I showed you in the trial that I'm going to be talking to you about are set up to say that given that you have all this choice in medicine, so we have 20 some antidepressants here, would it just be better to avoid the red bin medicines because we don't really know how to dose them, and the dosing could be a little bit tricky in that patient? And since you have a lot of choice, why not pick one of these where there's no genetic markers that we know would influence this particular patient? So it's been about choice of so a version of medicines that we don't really know how to dose. And that's the best way to think about it, not necessarily about the medicine that would work better for this patient. So let me show you the results of the guided trial. So this is, the first slide here is about what medicines were chosen after the time of randomization. Remember, all patients came in on some antidepressant. Half the group, the guided group, got the pharmacogenetic results back to the provider only. And what you'll see here is that for the vast majority of them, the patient got switched over to a medicine that was not a red level, not a red bin medicine. So essentially, at the end of 8 weeks, 91% of those patients have been switched to a medicine that was either in the green or yellow bin in this particular trial. So they sort of ignored the yellow bin. And then the arm that got the results back, there was no change in the sort of distribution of which medicines were red. It's a really important piece to this, and we are finding the exact same thing in prime care. About 75% to 80% of the time, the genetic results aren't helping you choose or avoid a red bin medicine or a highly genetically influenced medicine. So about 80% of the time, by chance, for a given patient, you're going to be choosing a medicine that wasn't influenced for that patient. So it depends on whether you're a glass half full or glass half empty kind of guy or person. 4 out of 5, is that good, or is 1 out of 5 good? I would suggest to you that from a public health perspective, if avoiding something that we don't know how to dose well leads to better outcomes 20% of the time, that would be a phenomenal public health impact, right? I would more than guess that 20% of the time that you get a cholesterol, it's uninformative. So I'm not sure that 80% is a bad number. That essentially the number needed to treat to be helpful here is 5. Is another way to think about the test. Now, did it matter? So here's the primary randomized outcomes in this trial. And they had actually a fairly odd finding, I think. The continuous variable, so these were, I want to say these were MADRS levels. No, they're HAM-D, sorry. HAM-D scores. The continuous measure of HAM-D scores actually wasn't different. It favored the guided trial, so the percent improvement was 11%, but that wasn't statistically significant. But interestingly, the dichotomous variables of response or remission both favored guided treatment. So in the primary analysis of this study, guided treatment was better than not guided treatment when it came to response and remission, but not symptom improvement. It's a little bit of a fluke typically that your continuous measure doesn't show something when your categorical measures do. It means there's a lot of people lumped right around the criteria of response and remission. And if you look at the raw data, that's actually what's true. I was not an investigator in this, although Penn was a participant. They also went back and looked at, did it matter, just forget randomization. So this was clearly a secondary post hoc analysis, but if you forget randomization and just look at whether people got better related to whether they ended up on a green Ben medicine or a red Ben medicine, then you see pretty whopping effects of difference. So the folks that end up on green Ben medicines are actually green or yellow for the guided trial. Even in the continuous measure, you get a 60% improvement, a 71% improvement in response, and 153% improvement in remission. This is not a randomized effect, it's a post hoc analysis, so take it as you will. So back to those 4 trials. 3 of the 4 trials have actually shown one of them is this trial. So 2 of the other 3 have shown very similar effects. That having a pharmacogenetic test panel result and avoiding medicines that might be challenging to dose because we don't know exactly how to dose them leads to clinically observable positive effects. The one trial that didn't is also the smallest. And from a pharmacogenetic study perspective, it's a tiny trial. Remember that 80%. So if 80% of these ended up on a green med anyway, then you're only talking about a trial that's really an N of 56 here. And so that Perez study is because 80% of the time, essentially the test probably was not informative. So this ends up being a pretty small trial. I'm not here to say that you guys should start ordering these tests right away, but it is encouraging that 3 of the 4 of these studies have shown effect. So with the last few minutes, I want to show you what we've been learning in prime care and tell you a little bit about the prime care study. So the VA decided that before we started doing pharmacogenetic testing broadly, and before we started spending millions of dollars in this space, we really needed to have some more evidence. And actually at the time, the guided trial, the time that the prime care study, which is the VA's trial, started, the guided trial was in progress, but the results were not known. So the VA funded a multi-site trial. You guys are a site. Amanda Wood is the local site investigator. I'm the overall PI. I think we started about 3 years ago together. The goal is to randomize 2,000 patients with major depression across, actually it's now 22 sites. Actually today it's 15 sites because we've let some sites go. But overall it was 22. And the goal is twofold. So and I'll show you the design of this in a second, or talk a little bit about the design. So the patients and the providers are randomly assigned to the intervention group, in which they receive the PGX panel right away at the time of a prescription change, or a delayed group, they get them back 6 months later. The outcomes are measured over 6 months by a centralized call center. So they're blinded to outcomes. The primary hypotheses are really twofold. One is that the providers and the patients in the intervention group will actually use the results. It will be used to guide their treatment choice. And the other is that mattered. So those veterans in the guided arm get better, or get better faster, however you want to think about it. And we have a whole series of secondary outcomes and discovery outcomes that be happy to talk to people about. The VA did decide, I actually don't think the VA's ever done a 2,000 person randomized trial in depression before. They've done some large studies, but so there's a fair amount of discovery that's been built into this, and we are getting serum samples at 4 weeks and DNA for whole genome, et cetera. So there's a fair amount of investment in this trial broadly. It's going to be a wonderful data set for a few years. These are the sites you'll see somewhere on here. I guess they're in alphabetical order. Puget Sound's on here. So thank you guys for participating. You've actually been the best recruitment site by far throughout the whole trial. There's clearly a lot of interest in pharmacogenetics out there. This trial was different than the other 4 trials. The other 4 trials were done as fairly standard RCTs. Even if they were multi-site, there would be a sort of a single investigator taking care of the patient or a small group of investigators at the site. So you would recruit through ads in the paper and so forth, and you would take on the care of that patient, just like a pharmaceutical trial. In this trial, we're doing it as a very pragmatic implementation trial. So we're doing it at point of care. So these are regular providers in the clinics. We are not taking their patients away from them. They are participants and they actually consent to be in the trial as well, the psychiatrist and the primary care doctors. They determine eligibility. So to make a referral, they actually give us a referral and say these 4 or 5 things, that this is a patient that has depression. I'm about to start an antidepressant. That could be a start or a switch, but it can't be an augment and it can't be a change in dose. That start is going to be monotherapy, and that they can't have schizophrenia or bipolar active SUD, and they're not in the hospital. So the provider actually determines eligibility for the most part, makes the referral. The research assistant locally does a PHQ-9. They have to have a 10 or more, and they have to be 18 to 80. That person is then consented and eligible. It's not a long baseline. The original idea before COVID was that the provider could make a referral and the patient could get randomized in an hour. So real point of care study. That's not happening now because of COVID, but that was happening pre-COVID. And the providers are the ones getting the test results back and interpreting them and talking to their own patients about the test results, et cetera. So we've learned a lot in doing this trial over the last 3 years, and one of them was about the providers. And the first thing we found out about the providers is they know squat about PGX testing. So on the left was a questionnaire we did at the beginning. So this was a questionnaire to the providers. How many of them knew that that the FDA has labeling information about pharmacogenetics? And 75% of them didn't know that drug labels had any pharmacogenetic information on them. And the panel on the right was about their past experience with ordering either disease genetics or pharmacogenetics. So the disease genetics was much more commonly done. So primary care doctors probably have ordered something for Huntington's or for Alzheimer's disease or something. Almost half of them had done that, but on some psychiatrists. But on the right side, only 8 and 15% had ever ordered a PGX panel. So out of the gate, we actually had to develop a whole infrastructure for educating providers. And so we created videos. Mike Thace and I did some videos of how you talk to your patient about this. We created videos that were like 2 minutes long that you could show a patient so that you could introduce pharmacogenetics to the patients, et cetera. So we spent a lot of effort. One of our cores in this study was really about the education of providers and to some extent patients. Some of the other learning lessons was, so besides that, and all those videos and stuff are actually publicly available because we're the VA and the government. We can't hide any of that stuff. You can just Google Visinfor, MyRec, and Prime Care, and you'll come to our website, or there it is. We also realized that we needed subject matter experts at every site. So we actually, the local site PIs became not only the PI of the trial, but had to learn how to be pharmacogenetic experts overnight. And we spent a lot of time. So Amanda Wood is that person for you guys at the VA. We basically trained Amanda to be not only good at doing the trial, but being the go-to person for answering questions. We had to figure out how to do point of care study. Doing it on the scale pretty much hadn't been done before. And we created an infrastructure to do this. So, and we've totally had to pivot with COVID, and I'll show you that we've actually successfully done that as well. So this is just recruitment. So recruitment is, so you guys are, 9 months early for me giving you results. So recruitment officially is to end at the end of January. Will we make 2,000? I don't know. We'll come damn close. So we may not cross the 2,000 barrier, but we will come pretty daggone close. So the blue line here, I think I'm colorblind, it was what we were supposed to do. You'll see there's an inflection point. So when COVID hit, we renegotiated with the funder, in this case, the VA, and extended recruitment by a couple months because of COVID. But we only asked for 2 months, and we should have asked for 4. You see the line got flat for COVID, but then we were able to totally turn this. So this was a completely face-to-face recruitment typical trial up until COVID hit. It is totally a virtual trial now. We send spit kits and cheek swab kits to the veterans' home. We've had almost no, knock on wood, almost no problems with doing this trial. You can actually see the slope of the recruitment is in some ways as good or even slightly better than it was pre-COVID. With that, we still are slated to end in January because that's when the funding for the sites ends. This is just, it's a while ago, but just who's getting into the trial. These are sick patients. So the demographic stuff on the left really shows it's a VA sample, a fairly large minority participation, which is great because almost all of the evidence of these markers is based on European Americans. A much higher rate of female participation than what's in the VA. My gut says that that's because affective disorders are typically more found in women than men, and the exclusion of substance abuse, which is more found in men than women. Not surprisingly, most of the patients are coming from mental health providers, but we actually, a third of them are coming from primary care. Their average PHQ-9 score at the beginnings is 17. So again, these are pretty sick patients. Lots of them have comorbid PTSD. Lots of them have anxiety symptoms. Some of them drink, some of them use marijuana. A few use other drugs, which means that they shouldn't have been in the trial, but that's actually 4% is good. And a quarter of them use tobacco. There was one other thought I had about this in terms of the patient population, but it went out of my head. So, just, it is a sick patient population. So one of the other learning lessons I just wanted to share with you is, again, the test utility. So that's actually so the 2 use cases for pharmacogenetics from a public health perspective are the following. One would be sort of your primary care patient that's brand new, never been on an antidepressant, and you wanna pick the right medicine from the beginning so that they never become treatment resistant or avoid the medicines that are going to be more complicated for that patient or lead to more side effects. So from a public health perspective, if we did that a little bit better, it would really drive the cost down. Jürgen would be very excited. The flip side is the way most psychiatrists think about this, and that's that I've got complicated patients that I'm not even sure if they have depression. They've been on 15 drugs over the last 20 years, and would this help me pick the next drug for them? And probably both use cases have their face validity, right? We wanted to see a little bit about what happens in the trial. So without breaking the blind, we did the following. We wanted to look at a lot of the patients that were enrolling actually were on psychotropics at the time of enrollment. So they were patients that were coming into the trial to be switched. So we asked the question, in those patients that are currently taking an antidepressant, what proportion of them were taking a medicine that would, by this company's algorithm, was classified as having a high gene-drug interaction potential? The other thing that we're doing in the trial, and we've not published on this yet, well, this paper, what the results I'm about to show you is in press at this point, is at the time of randomization, we're also asking the patient, the provider, before they get the pharmacogenetic results back, before the patient's actually randomized, what is your intended med that you're about to use? So what are you going to switch to? Or if it's a patient that's not on something, what medicine would you be recommending for this patient? So that's the next intended medicine. Remember, they don't have the PGX results back. That's a totally, what is their clinical impression of this patient? What medicine would they typically use next in that? And what we did is looked at in these actual patients, their genotype results and sort of what that shows. And what you'll see here, so look at the graph on the left first. So those veterans that were on an antidepressant, 20% of the time they were on a medicine that was in the red band. And if moving them from a red bin medicine to a green or yellow bin medicine is clinically useful, what this suggests is that 20% of the time, that would be a smart thing to do. That's a pretty good hit rate for a lab helping you in terms of taking care of somebody. And then the same thing for next intended. It's about 18%, it's basically 20% as well. This is almost exactly the same numbers as what the guided trial found, interestingly enough. So it was 76%, I think, or 24% in the red for them, and we're hitting at 18%. I actually think from a public health perspective, if we continue to show good clinical outcomes from avoiding those red bin medicines, that this is actually not a bad result. So the question about new versus old, it is actually true in this sample that if you have a past history of having other treatment trials, of having multiple treatment trials, that the chance of the next treatment being a red bin medicine or genetically influenced medicine is higher. So it does suggest that there's a slightly better utility, an odds ratio of 1.6, of using the test or having the test be meaningful in patients that have sort of treatment resistance. And that's where we are. So I want to thank you. I think the punchlines, and I'd be happy to, we've got some time for some questions. The punchlines for me are the following. We do not have pharmacogenetic tests right now that tell you which medicine is the right medicine for a patient. We just aren't there yet. We may be there in 5 years, maybe there in 2 years, but the currently available literature does not tell you which medicine to pick for a patient. It might actually be that we can tell you which medicine to avoid in a patient, and avoid only in the sense that in those patients that have genes that are influencing the metabolism for that patient that we don't really know how to dose well in that patient. So given that we have so many choices, why not just avoid that? And it looks like, at least from 3 of the 4 published trials, that avoidance of those medicines actually leads to better outcomes. And we'll see within a year if that's the case for the VA's big study. So that's the punchline for me. So with that, I'm happy to address any of the questions that might have accrued.
Thank you, Dr. Oslin. And I'd like to remind others to...
I'm going to stop sharing, right?
Sure. Please write questions into the chat or into the Q&A. As questions are coming in, I thought I'd ask one to get us started. As we're waiting for the results over the next year or so, I'm thinking about scenarios where I commonly see these test results, which is either in primary care record that I'm reviewing or patients bringing in results that I see. How might you advise a psychiatrist at this point to handle a circumstance like that, having the results that maybe the indication for ordering isn't clear to the clinician who's seeing those results?
Yeah, no, it's a great question. So what do we do kind of now? So you've got 3 or 4 trials that actually show that avoiding medicines in the red bin, or whatever company it is, they're comparable red bin. So some of them don't color code in that way, but 3 of the 4 trials suggest that that's not actually a bad thing to do. And with so many choices, what little, you have little to lose, right? So the question, if the results have been already obtained for a given individual, you have little to lose in using those results and choosing a medicine, right? Um, I will tell you the other factor in that is that our early experience with this with patients is that they read these reports and they overinterpret them to a T. So they totally read these reports that red is bad, green is good, green means I'm going to respond to it. Now you can think of that in two ways. And interestingly, in the survey, I'll tell you which way the survey went when we asked providers about this. But you can interpret that, oh, here's a crackpot result that's overemphasizing a result for a patient, and that they're just missing it and they don't understand the science, and they've put too much value on this. The other way you can interpret is, okay, so there's a little bit of a placebo effect from reading this shiny piece of paper that says that the green med is good for me. And maybe that's not such a bad thing. Placebos work, right? And I will actually, what was interesting is we sort of asked this question in the survey in a roundabout way, and providers were totally cool and comfortable with the placebo effect, and thought of that as a positive. So you have two factors. Now, the real question is, should you go out and start, ordering one of these things on every single person? They're not cheap. They're commercial prices in the $2,000 neighborhood. I'm not sure. So there are clearly institutions. I actually have no idea where you guys are. There are institutions that do this on every patient. So the Mayo is a good example. When you go to the Mayo, you get a battery like this as a new patient. It goes in your record and it's there permanently. So I would say there's a couple things that are interesting here. One is that these cytochrome P450 enzymes, they're not unique to psychiatry, right? So the informatics challenge isn't so much, I get a result back and it tells me something about sertraline, is that I get a result back and it actually tells me something about 50 or 60 different med classes, right? It's not a unique result to psychiatry. It's telling me very broadly about a lot of things, and some much more salient than an antidepressant. So the informatics challenge is if you as a psychiatrist order this lab test and it comes back that this person's a 2D6 rapid metabolizer, how do I extrapolate that to the 10 other medicines the patient's on, and feed that information back to the primary care doctor and the cardiologist and all the other providers that are taking care of patients? And that we're still struggling with that in large ways, particularly in big systems like ours and yours and so forth. I'm not personally ready to start ordering this test on everybody, but we're close. I was a total skeptic going into this trial. I will have to tell you that. I took on this challenge of doing this trial not because I thought that this was, well, so I think genetics is in our future, right? As mental health providers, but I went into this thinking that we would just show this thing not to work, and I've gotten totally intrigued by it. I mean, 20% is not bad.
A new question in the chat. What if there's individuals getting treatment with multiple medications, which can complicate interpreting these results? Would you consider obtaining drug levels if patients are having non-response or side effects at a usual dose?
It's a great question. So none of these trials have been done in people with multiple medicines, although we are about to start a trial with a slightly different question. So for me, polypharmacies, we rarely get much out of polypharmacy, right? So we get something out of simple augmentation in terms of better responses, but a lot of these patients end up on 3, 4, and 5 psychotropics, right? They accumulate psychotropics. We're about to start a trial in which it's a small pilot, so I don't know that it'll ever make a big hay in the literature, but we're going to take patients that are on polypharmacy and get testing or not get testing back to the provider with the idea that it might actually help the provider take that patient off of some medicines that they're on, right? Because it's always complicated for us to take medicines away. And to see if that then leads to actually better outcomes. That comes from my geriatric training, where I spent the early parts of my years as a geriatric psychiatrist taking more patients off of medicines because they sort of accumulate so many. And so that's an interesting concept. The idea of drug-drug gene interactions, it is in the market. The reason that hasn't penetrated well has to do with the machinery of this. Remember, these are lab companies, so they actually get no information about the patient. So they don't know about the medicines that a patient's on. So you're sending a sample to a lab company and getting a test result back. And so they wouldn't know if there are drug-drug gene interactions, right? Because they don't know the medicines that the patient's on. But there are informatics platforms that are starting to think about this. How can you combine, all of the meds that somebody's on with their genotype and have some type of interpretation around that array of medicines, and not just psychotropics, but all the other medicines that are metabolized.
And based on
and the drug, I can just answer the drug level question. Drug levels don't have a very good history in our field, right? So, except for in select cases, some of the TCAs and lithium and a few very select cases. One of the discovery pieces to this, so all of this hinges on metabolism. So the real question is, how much has metabolism really been affected by the cytochrome P450 system in a clinical way, right? I don't know that we'll be able to fully answer that, but we are testing serum drug levels for the antidepressant that they're on at 4 weeks in the trial to see if we can back into whether these genotypes actually predicted dose. It's going to be a very tricky analysis. 2000 may not even be close to enough to do this in, because these patients are on all different kinds of medicines. But maybe there'll be enough sertralines, for instance, or something, or citaloprams that we could say something about a particular medicine. And that would still be an advancement to the field, because our field doesn't have good outcomes with doses.
And regarding the circumstance with a patient on multiple medications, I mean, you've commented on the red box or the green box being interpreted as effectiveness for that individual. Do you think that a red box or green box medication might make a patient more interested in stopping a medication in the circumstance of a multiple medication?
Yeah, absolutely. No, the patients totally misinterpret these results. It's very clear to me, even with some good education. Most of them totally get that this is about the right medicine for them. And to be honest, a lot of providers don't understand that as well. Nobody that heard this talk today, but I can tell you that a lot of providers see from whichever company get these results back, and they say, oh, this result says this medicine is going to work better for that patient. And that's totally how they interpret it. They totally miss the science to it. So that concept is sort of perpetuated by people in our own field.
Okay, great. Well, we're just getting to 1 o'clock. And thank you to Dr. Oslin for presenting today and walking us through the field, and for presenting on the Prime Care study. And we'll look forward to seeing results over the next 9 to 12 months. So Thank you everyone for joining us.
Thank you guys. Have a good afternoon.